Alright, so we're just gonna begin the section A module 2 question for the 2017 biology paper, and it asks us, well, it says that hemophilia is a recessive sex-linked condition in humans in which blood does not clot properly, leading to the excess bleeding. The use of the symbols XH for normal allele and X common H for the hemophilic allele, right? We're supposed to be using these things in our responses. Now, if we state the genotype, for normal clotting male, we're gonna have the X chromosome with the normal X chromosome and the Y chromosome, right? That looks weird. That looks like an X. Can't work. Gonna have the Y chromosome here, right? So males are XY, and then we're gonna have a normal carrier female would be heterozygous, right? Right? So that's the female carrier. Now we're asked to construct a Punnett square diagram to show how hemophily affected offspring can result from the normal clotting parents. Right? So that means that the female must be a carrier. So if we look at a normal clotting male, right, and we look at a carrier female, which is also normal clotting, right? We're gonna be getting these derivatives here. So we're gonna get a homozygous dominant female, a fine male species there, all male member here. Alright, we're gonna end up with another carrier female and a hemophilic male, 25% chance to get that hemophilic male there. Right, so give an explanation as to why a man with hemophilia cannot pass the condition to his son. Within males, males pass the Y chromosomes on to the offspring if it is going to be a male. Therefore, since the characteristic is sex linked upon the X chromosome, right? And men can only give the Y chromosomes to their son, right? Then hemophilia cannot pass from a male to his son. Right? That should make sense. It's sex linked on the X, and if it's going to be a son, the Y chromosome will be passed on, therefore not carrying the gene because it is sex linked to the X chromosome. Lovely. Right? Everything there is supposed to be lovely. Now a geneticist, right, crossed the two types of P that's obtained to 5,474 plants with round seeds and 1850 plants with wrinkled seeds for the second filial generation. You are required to use a chi-square test to show the results and that the results are consistent with the 3 to 1 ratio normally expected in the monohybrid cross. So what is the null hypothesis? Again, the null hypothesis would state that there's no significant variation, right, between the two characteristics, between the two alleles, right, being expressed, and that any significant change would be due to chance. So we know that is the null hypothesis, and now we look at the chi-square test specifically, right? We need the observed and the expected, right? So we observe 5,474 round seeds, right? We also observed 1850 there for wrinkled seed. And for the expected, now what we're going to be is combining the two values, right, to get our total first. So we plus them together, 1850. Right, so the total itself is going to be just adding the two values together to get 7,324, right? And what we need to do is now establish the 3 to 1 ratio. Therefore, one way in which we do it, and I'm gonna use the same way that somebody proposed yesterday, that we're going to just divide everything by 4 in order to get one part, right? So if we divide it by 4, right, so divide everything by 4, so 700, well, 7,324 divided by 4 should give us 1831. Right, so 1,831. So if we look at a 3-to-1 ratio now, in order to get the third ratio with the round seeds, right, we're gonna have 1831 times 3, and that should give us, times that by 3, that should give us 5,493, right, for round seeds, and for the wrinkled seeds, um wrinkled seeds, right, we're going to have just 1831 there, right? So with that information now, we start plugging in our values. So our expected now would be 5,493 for a round, and it would just be 1831 for the wrinkle there, right? um Looking at the observed minus the observed minus the expected, here we're supposed to be getting negative 19, and down here we're supposed to be getting positive 19, right? Would that be it? And then we square it. Would that be it? Let me check, right? All right, so it would be 5574, and we're going to be minusing 5493. Observed minus the expected. Looks like there's an issue here. All right, what are the values that should come out here? Let me just remove this. The values that should come out here, or the values that we were getting before. Guys, what were the values that we were getting for yesterday? That actually was in the call yesterday. Nobody? Speaking directly to you guys. So we're gonna get 19 down here at the top of what are we supposed to get?
Negative 19.
Alright. But is that the value we get when we minus the expected from the observed? You don't get negative 19 when we minus those two values. So is it that there's an issue with the calculation itself that we got there? That's intriguing. Let me just do over the calculation there to make sure that this makes sense. And then we divide it by 4, and then we times it by 3. That is the value. So is it that I'm doing something incorrectly with the calculator? So that would be hard to believe, because I'm getting 81. So where we got the negative 19 from? Look at that, I put the wrong value here. Right, so this is the wrong value here. I put 5 instead of a 4. All right, so that is where the wrong value is coming from, negative 19. All right, lovely. All right, and when we square negative 19, what we're going to get? We're gonna get 361, right? And then that would mean, and then what have here 361. We're gonna get the realm, if we square anything, right, based on the function, it's going to give us any positive integer. So the negative values in this range don't matter. And then we know to start to divide the 361 by the expected. So 361 by 5493 would give us 0.066, right? And here we're going to get a value of 61 divided by 1831, right? That would give us 0.197, right? So look at 0.197, right? All we're going to do now is 0.066 plus 0.197, right? That should give us 0.263. That is our chi-square statistic, right? So using our chi-square statistic now, we're referencing the data, right? We're trying to create an inference, right? So before we really look at that now, well, let's just look at it, right? So in order to get the chi-squared value, we need to declare our degree of freedom, and because we're only working with round seeds and wrinkle seeds, that means our um number of classes is 2, and in order to get the degree of freedom, we minus 1 from any number of classes to get 1. So our degree of freedom is 1. So look at this range. The critical value generally tends to be 5% or 0.05 for the probability value. So we're looking at here, right? Our actual value for the degree of freedom 1, our critical value would be 3.84. And that means that if our value exceeds 3.84, therefore we would have to reject the null hypothesis, right? Because our value is 0.263 and beneath this value of 3.84, then we will have to accept the null hypothesis and state that there are no significant variations between round and wrinkle seeds, and any variation is due to chance. All right, that should be lovely. That should be fine, right? This asks now, well, gives us a size of an organism in a population, right? And we're asked to look at the effect, illustrate the effect of disruptive selection. And we know disruptive selection would have a selective pressure against the average size of the organism and select for two extremes, right? So disruptive variation will look like this as the selective pressure goes against the average, right? So it's going to select in favor of two extremes. And that is what we're going to have for a disruptive selection graph there, right? Using Darwin's theory of natural selection, explain how disruptive selection can lead to the formation of new species. So first, we're looking at Darwin's theory of natural selection, stating that we're basically speaking about the survival of the fittest. That when organisms themselves adapt to obtain advantageous traits, right, they're more likely to live towards maturity and to carry on those advantageous genes, right, to the next filial generations. Therefore, right, that would have the continuity, right, of the most advantageous genes within the specific environment. If we have a selective pressure selecting against the average, right, size of an organism, for example, what was above there, right, selecting against the average trait, we're gonna disrupt the traits within the population, right, favoring the two extremes, right? And we know that natural selection, right, is time based. So if these two extremes are to be selected for over a long period of time, this may result in some amount of speciation, the divergence, right, of two separate groups within one species that in time becomes two separate species. We're going to be speaking about how that disruptive selection can lead to the split within a species, right, as the different traits complexify over time, resulting in organisms that no longer have the ability to sexually reproduce to produce fertile offspring and viable offspring. So we're just looking at that concept there for this question, right? Shouldn't be a problem. And this now is just looking at a graph question interpretation of the graph and looking at the germination, right, of pollen within two different types of culture media. So we're going to be drawing general graphs and distinguishing the curve, right? So it's going to be, yeah, so we'll plot line graphs, right? So now with line graphs, right, we're gonna have to distinguish the two curves, right? So if we're gonna be looking at the liquid culture medium, we could use dots and circles, right, in order to plot those points, and then we could use x's to plot the solid culture points. You know, you just have to differentiate between the two curves, right? And then we're gonna be looking at the conclusions that we can draw, right, from the data specifically, because I'm on a digital platform, I won't be able to like draw the graphs properly. So we're supposed to describe the overall trend and the effect of the different source of sucrose concentrations in both culture media on the germination rate in pollen. Alright. And we're looking at how the type of culture media would actually affect the maximum concentration. Right? So we're looking at two curves, right? So if we look at the first curve, let's say that the first curve is like this, and the second curve would be something like this, right? So this would be probably for the liquid culture, and this would be probably for the solid culture, right? So we're looking at both the fact that both of them will steadily increase as the sucrose concentration increase, right? And then start to fall off after a certain point, right? So both of them have their maximum at around 40 for the concentration there, right? Both of them have a maximum of around 40 for their concentration. Right, so you guys just have to plot the graphs in order to make that known. Right, and we're looking at the fact that the general trend is that they increase as sucrose concentration increase, the percentage germination increases as the sucrose concentration increases, and then continues to fall off after maximum point is reached at 40 grams per liter. Right, we're looking at that. And then we could also state that the culture media, right, it looks as if within the culture media, the initial rate of pollen germination or initial percentage of pollen germination seems to be a bit slower than within the liquid media, right? But overall, the solid culture would produce a higher content or a higher percentage of pollen germination than the liquid culture media, right? And we could look at the effect of that the solid type culture medium on the liquid culture, well, on the pollen germination, right, in relation to liquid culture medium, right? So the solid culture medium allows for the percentage increase to be a little bit slower as sucrose concentration increases, and then it reaches a higher maximum than that within the liquid culture medium. And with the liquid culture medium, we know that the increase is much faster, right, towards a lower maximum than what the solid culture medium would produce, right? So it's not, we don't even really have to apply any information about germinations of pollen here. It's just using graphs and speaking about that structure there. We've done this as well. For one, we've stated that this would be the chorionic villi. Two, we said that two would be the endometrium, and we see that three would be the umbilical cord, right? Through much discussion and debates yesterday, right? So the structure in one, right? The function of the chorionic villi would be to actually to have the absorption of nutrients along with any other type of useful material from maternal blood supply towards the fetus. So the chorionic villi is there within the intervillous space as the space between the placenta and the fetal blood supply. So we're looking at the fact that the chorionic villi is used to absorb any nutrients and useful material from the maternal blood supply so it can enter the fetus. Two, if we're looking at the endometrium for two, right, then we're looking at the fact that endometrium would provide us a suitable environment for fetal development. That's generally the main function there. And for three, if we say that three is the umbilical cord, it would be the main passage through which blood can enter into the fetus. So we're looking at the fact that it provides nutrient maximum nutrient movement into the fetus, gaseous exchange, and removal of waste. Right, so we're looking at generally those functions for those three structures there. Right, our comment on the two rows of structure Y. Structured Y is labeled as the amniotic fluid. You know the amniotic fluid is there for shock resistance to protect the child. Right, it provides suitable temperature regulation, right, for the fetus, right, as well as it provides some amount of it helps the fetal with the development of swallowing reflexes as well. So we could look at that. Mm-hmm. All right. So we could look at those things that general uses for what is labeled in Y. Right. And now we're asked to outline two ways in which the placenta acts as a protective barrier for the developing fetus. Right. So first of all, we know that the placenta really creates a barrier between the maternal blood supply, right, and the fetal blood supply. Right, so two ways in which is a protective barrier, right? The placenta actually doesn't allow for certain viral or pathogenic components to pass through, right, into the fetal blood supply, right? So it does not allow certain, that word is important, right? A certain harmful material to pass through the maternal blood supply into the fetus, right? It allows a pathway for the blood pressure to decrease going to the fetus because the fetal blood pressure is going to be much lower than the maternal blood pressure, right? It's going to be providing also it's a protective barrier, right? Because not only does it protect the fetus from any type of pathogenic, well, from some pathogenic components, right? But it provides some amount of passive resistance, right? And antibodies towards the child, right? Well, not the child, the fetus. There's a difference, right? Towards the fetus, right? To assist in fetal protection while in embryonic development, right? So those general things can be covered there from those two sections. So this is really the module 2 and module 3 section, right, for the 2017, right, for section A.